Doctor Omar Pantoja.
Yep, we seem to have lost.
Yeah, because I called. So we'll start again. Okay. Perdón, ahora sí ya, otra vez. Bueno, gracias por estar aquí. Es un placer tener a John Cushman, profesor John Cushman de la Universidad de Nevada en Reno. John ha trabajado casi toda su carrera con plantas CAM, del tema del que nos va a platicar hoy. Él obtuvo su universidad, o hizo la Universidad de Neusiedl College en Pensilvania, después hizo su maestría y doctorado en la Universidad de Rutgers y un postdoctorado en la Universidad de Arizona con Hans Bonner, para eventualmente moverse a Oklahoma State University como profesor asistente, donde pasó aproximadamente 10 años, y después se cambió a la Universidad de Nevada en Reno, donde está desde el 2000 aproximadamente. Ha estado investigando sobre la productividad y las respuestas a la irrigación de plantas CAM muy productivas, como son la tuna, para estudiarlas como fuente de alimento y también como fuente para biocombustibles. Recientemente ha usado ingeniería de tejidos suculentos para mejorar el uso del agua en plantas no CAM, y tema del cual nos va a platicar el día de hoy. So John, thank you very much for being here, and it will be good to hear your research on this subject, and welcome to Cuernavaca.
Great. All right, thank you. And let me just go ahead and start my share. There we go. And thank you, Professor Pantoja, and welcome everyone. I really appreciate the kind introduction and the opportunity to speak with you today. And I'd like to send my regrets for not being able to visit in person, but we have Zoom now, and so I'd like to share with you my research on some of the novel strategies that we've been taking to improve water use efficiency for a hotter, drier world. And for those of you who are not familiar with my research program, this is just an overview of some of the projects that we're working on. And we live in Nevada, which is the driest state in the United States. And so we work on developing alternative crops like oilseed crops and cereal crops. But I'm going to tell you a little bit about our work on cactus pear before I get into our work on crassulacean acid metabolism. And we'll talk about our efforts today on synthetic CAM and tissue succulence engineering. And we've also worked in collaboration with researchers in Panama at the Smithsonian Tropical Research Institute on CAM evolution in orchids. And so just real quickly, we have global climate crisis upon us. In addition to COVID-19, we have global warming. And so we have a dry land expansion that's expected to add to this 42% of the Earth's surface that is already arid. And these arid lands, which are shown here, have an important role in the entire global ecosystem. And there's some facts here, mainly that dry lands produce 67% of the world's food. And that's really quite amazing. And 84% of the world's cultivated area. Now, much of these areas, if you're going to grow crops, they must be irrigated. And so this brings up situations where you're faced with having to improve drought and salt tolerance for those crops. And those are two issues that we're hoping to address through our research. So just to give you another snapshot into global warming, you can see over the last 40 years that just in the United States, we see very high increases in heating, shown here with these temperature increases. And coupled with that, we have, we've always seen a lot of variation in precipitation patterns, but in the Western US, over the last 8 to 10 years, things are getting much drier. And the prediction is that over the next 30 to 50 years, the Western US is going to get drier and drier. And this is going to add to a situation where we are already very dry, and we are experiencing the consequences of global warming now. So we've had tremendous wildfires near us, and so Reno is right here where my red dot is, and so we've suffered a lot with smoke and wildfire damage this year. And this is also compounded by the loss of our water resources. This is a reservoir north of here, north of Reno, where they've had both wildfires and a loss of that reservoir capacity. So the prediction is that with warming, you're going to get increased drying, and soil aridification is going to increase for many areas, and this is going to force soil drying. And the ability of a plant to compensate through elevated CO2 that are also part of global warming is going to be inadequate. Plants are not going to be able to compensate fully for these drying effects. And this is going to end up causing decreases in crop yields. And so this is just a projection going out over the next 50 years or so, just showing you that 50 to 80 years, just to show that crop yields are going to get worse and worse if these drying trends increase. And the calculation is something on the order of a 1% loss in yield increases for every 1 degree C of temperature rise. And this is just looking at these 4 major crops. So with that, this brings us to our research on CAM. And this is just to show you that there are 3 major forms of photosynthesis. So we have C3, C4, and CAM. And these are just some representative species. And what CAM does to improve its water use efficiency is, like C4, it does a primary, these plants will conduct a primary carboxylation reaction bicarbonate using the enzyme phosphoenolpyruvate carboxylase. And this causes these plants to accumulate malic acid at night. They use that malic acid to decarboxylate, and they use a series of enzymes, malic enzyme, or an enzyme called PCK, to release the CO2. That CO2 release is refixed through the Calvin cycle and Rubisco. And this primary fixation happens at night when evaporative water losses are lower. And so this ends up with, for the plants tend to be more water use efficient. So just to rephrase this a little bit, most of the CO2, not all, it depends on the plant, but most of the CO2 is fixed at night when these evaporative transpiration water losses are low. And this improves the overall water use efficiency of the plant. And then during the day, this CO2 is released and refixed by Rubisco behind closed stomata. So stomata are open in phase 1 and closed for all of phase 2. And then sometimes they reopen it. This is a very plastic adaptation, but the main point here is stomatal closure during the day is going to improve your water use efficiency. So with that, and they accumulate malic acid at night, and they degrade storage glucans during the night, and during the day, these two are reciprocated. So you have this diel flux of malic acid, and storage glucans. And this is going to become very important later on. I'm going to show you a slide that looks very much like this increase in malic acid. So just remember this increase in malic acid and this reciprocal decline in storage glucans. So the net result of CAM, you can sort of compare it to these other crops that they use about 20% of the water. And the productivity is lower for CAM, but there are certain CAM species where the productivity can be quite high. And this is just a summary of a number of different high performing species of C3, C4, and CAM. And you can see that these CAM species, mainly agave and Opuntia, perform quite well. And so we got very interested in this. And for the last 6 or 8 years now, we've been working on Opuntia. Obviously, Opuntia, the epicenter of diversity of Opuntia, it comes from Mexico. And so, it has moved into the United States, and it can be grown along the southern tier of the United States. And this is going to obviously increase as things get warmer. But the biomass productivity is quite high. And of course, we can use Opuntia for fresh fruits and vegetables, biofuels, and many, many other various uses. And our interest is in using this for either ethanol or biomethane production. And as this is already being done and being fully exploited, well, not fully, but there are some major projects already going on in Mexico that are proving the concept that Opuntia can be a very great source of biogas. So with global warming, this purple area, these are some climatic projections that were done by Nick Owen a few years ago. And it's just showing this purple area for agave ticulana and agave Opuntia. As things get warmer, where we can grow Opuntia in the US is going to increase. And these modeling predictions demonstrate that Opuntia is going to be more climate resilient overall than agave. And so we focused our interest on Opuntia. And with that, we did a 5-year field trial in southern Nevada and near Las Vegas. And we showed that Opuntia is indeed has a very high biomass productivity and low water inputs. This is annualized water inputs of only 417. And we tested 3 different irrigation regimes, and you can see that if you give them 3, 4, or 700 millimeters, the highest water use efficiency occurs right around 400 millimeters. And we tested 3 different species. All the species have performed pretty comparably. These are just shown here with their fruit. And Ficus indica, of course, is the commercial variety, as well as Coccinifera. Both of these make a really nice, high-quality fruit, and they're very high performance biomass producers. And I guess the important thing to point out here is by year 5, this is being a perennial species, biomass productivity was up to 68 megagrams of dry matter per hectare per year in year 5, which is just an amazing amount of productivity for this crop. And it outperformed agave Americana by 10%, and it ended up using about 25% less water. So we're very happy with these results, and I think we've shown that Opuntia is worth investing in further. And so we are continuing this work with a grant from the USDA, the Department of Agriculture here in the US. And this is a picture of our field site in Parlier, California. This is near Fresno, California. And we're testing 14 additional high-performing species in this field site. And we're in year 3 of this project, and we're going to be assessing which of these accessions are really high-performing. And what's shown here is part of the National Germplasm Collection. We have over about 270 different accessions. Most of them have been collected in Mexico, Chile, Peru, South America, and we're evaluating these further. And if you were interested in understanding biomass productivity, but also things that are related to production barrier limitations. And so you can check out our project. We made 3 very nice videos. And if you go to this website, and also you can simply type in UNR and Opuntia, and you'll find these videos. They're on YouTube, and learn more about our project. And then one last thing about the Opuntia, the project, is that we also have been investigating ways of degrading this biomass for either ethanol or biogas production. And we recently characterized a soil microbial consortium, and we purified different bacterial strains. And then we did molecular barcoding to identify these species. And we also did digestion assays. And it turns out that one particular bacterium called Pectidobacterium cactusidia was very efficient at degrading these cladodes, and it secreted pectinase activity. So refer you to this work in Helion here for more information. And we are continuing this. This was done under aerobic conditions, and we're repeating this with microaerophilic conditions as well to see if these organisms perform under those conditions for biogas production. So with that, let's get into some more molecular studies that we've been doing on these CAM plants. And so this is an updated version of a figure that we published several years ago now, looking at all of the major either genome or transcriptome research activities that have been going on in CAM plants. And of course, the major models include orchids, agave, bromeliads, pineapple, not surprising, kalanchoe, clusia, which is one of the few tree species that conducts CAM. Our ice plant project, as Omar mentioned, and the Cactaceae. And so a number of Cactaceae species have now been sequenced, and I've just listed those. And our lab just finished sequencing Coccinifera, and we have an ongoing project to sequence Ficus indica. And as part of this large germplasm collection that we have available to us through the USDA in Parlier, we've surveyed these for their ploidy. And it's estimated that about 60% of Opuntia species have undergone some degree of polyploidization. And this is just a graph showing you the nuclear DNA content of these species. And so here we start out with a diploid species. This is another Coccinifera species, but it's a diploid, different from the one that I showed earlier. And then there's probably tetraploid species in here, hexaploid in here, and then we go to Ficus indica, the species that I showed you. These are all octoploid, and then we go all the way up to a 20-fold polyploidization, which is one species, which is just amazing. Instead of having, we go from 2, 4, 8 to 20 pairs of chromosomes, which is just an amazing amount of polyploidization. That's a bit much from a genomics perspective. And one of the things that surprised us is that this was a continuous range of genome sizes. We thought that there would be discrete steps, but we didn't really see that. And therefore, this is quite interesting to us and to learn more about what's going on with these genome structures. But to start out, we started with the simplest genome, and that was from this Opuntia coccinifera. So we've sequenced this genome. Now it has 22 chromosomes, and the genome is quite large. It's for a diploid species, it's 1.09 gigabases. And we're in the process of finalizing those data as well. And this is just a picture of what this species looks like with the beautiful flowers and the chromosome count of 11 chromosomes here. One of the things that we're doing in this project of the germplasm collection is we're sequencing the genomes, and the purpose of this is to get a molecular phylogeny. Genome sequences are very good for this, and so that is something that's in progress. And the genome, what's interesting is the genomes within this germplasm collection are quite conserved. You can see there's not a large size range of these different genomes, and this is just, again, this diploid coccinifera genome shown here. And it's really amazing. This germplasm collection is genetically quite homogeneous, and so one of the things that we're trying to do is to sort out exactly how many duplications are in this collection, and preliminary data suggests that there's quite a lot of genetic redundancy, and that way one of the things that we hope to do is to remove all of the redundant accessions so that we don't have to maintain them. All right, so moving on, we also have sequenced the ice plant, and so let's look at these two species. So we've sequenced in collaboration with Oak Ridge National Labs, Kalanchoe, and ice plant, and these were part of the Joint Genome Institute community sequencing program flagship plant species. So these were nominated in 2017, and we've actually sequenced Fanchankoi and Laxiflora. This is a diploid, and this is a tetraploid species of Kalanchoe. And then we will be adding ice plant to this very shortly. And in comparison, you can see these genomes are quite small, 246 and 390, compared to the Opuntia genome, which comes in at over 1 gigabase. But ice plant we've worked on for a very long time, and I think you're probably all familiar with this model. This is a facultative CAM plant, and when you water deficit stress this plant, you can induce CAM, and this can be done with either drought or salinity stress. And so this is very handy. It's very convenient because this allows us to pick out the genes that are involved in CAM, but many other stress adaptations. So this is the genome of ice plant, as I mentioned. It has 9 chromosomes, and it's about 370 megabases, as I mentioned, about 25,000 genes. And we've done extensive transcriptome profiling comparing the C3 state and the CAM state. And this is just some data that just to show you what these data look like on a large scale. We've done dial time courses looking at the fluctuation in expression patterns, and we've been able to find out that many thousands of genes are induced during CAM induction. And this is just a heat map of the C3 state and then the CAM state. And you can see in red the many genes that are induced. And you can see that there are some really clear diel and also circadian expression patterns as we move over this 48-hour time period. And so this is just to illustrate that this is what we've used to pick out the CAM genes. And so with that, this transcriptome data, we were able to pick out among the important gene family members all of the individual genes that are important to CAM, and these are just summarized here. So we have at night, we have beta-carbonic anhydrase, pep-carboxylase with its regulatory kinase, PPCK, malate dehydrogenase, and then this malate is stored, it's transported into the vacuole with several malate transporters. And then during the day, when the stomata are closed, this process is reversed. So we get CO2 release through malic enzyme. The pyruvate is regenerated through PBDK and its regulatory protein shown here. But you can also decarboxylate malate with malate dehydrogenase to oxaloacetate. and PepCK, and then PEP, that restores PEP, which goes back into gluconeogenesis and starch formation. So we were able to sort out all of the important genes that are able to do this carboxylation module and decarboxylation module, and work with these genes to try to generate a synthetic version of CAM. So this brings us to our synthetic CAM project. And so our question is, can we use a synthetic form of CAM in addition to tissue succulence engineering? And I'll explain that work in a minute. Can we use that either individually or in combination to improve the productivity, water use efficiency, and drought tolerance in Arabidopsis, which is shown down here? But also, ultimately, our goal is to put this into a crop plant, and we've chosen soybean as our target, simply because soybean is the most important C3 crop in the United States, but also in many other parts of the world, like Brazil and Argentina. And it's also susceptible to drought. It suffers many billions of dollars of crop losses to drought every year. So basically, what we'd like to do is install this carboxylation module that I showed you, the decarboxylation module, and then the two together, and see what effect that might have. And if this concept works, ultimately move this into soybean. So in order to do this, as I mentioned, CAM is controlled by a diel rhythm or a circadian rhythm, depending on the gene. And so we needed promoters to drive the appropriate temporal expression of these CAM genes. And so what we did was a data mining process that looked at things like circadian rhythmicity, drought inducibility, and their ability to be expressed in the mesophyll, which is where CAM occurs. So all of that work distilled down thousands of genes into a very small subset of appropriate candidate promoters that we could use to express CAM genes appropriately, the appropriate time of day or night, morning, day, or night, and then with the appropriate mesophyll expression. And we wanted them to be drought inducible. And the reason for this is that you don't want to perform CAM unless you need to, because there is a small 10% energetic tax to perform CAM. But CAM plants are happy to pay that tax because usually they live in very high light environments, but not all crop plants do. And so we thought it wise to also have CAM be drought inducible. So we had a lot of work to do. And so this was work by postdocs, very talented postdocs in my lab, Dr. Yim and Dr. Lim. And what they did was they mined these circadian data sets. They mined drought-inducible data sets, and they mined the tissue-specific expression of these genes. And what we ended up with is to test a series of 14 promoters for mesophyll-specific expression, and these were validated as shown here. And with then that, we have our genes, we have our promoters and terminators, and we were able to then make multi-gene assemblies of these genes. And for this, we developed a vector system based on work done by Gouillet et al. In 2013. We developed a position vector system, an adapter, and a carrier vector system. And we're working on a manuscript for the system, but basically it allows you to use gateway cloning and Gibson isothermal assembly to generate a series of transcription units or gene cassettes, put these together and assemble these multi-gene cassettes into gene circuits. So this is a picture of doing this. So this is a multi-gene assembly using gateway. So this is a single transcription unit being assembled. These then can be combined using Gibson assembly, and you can create a gene circuit. And this is just an example of one gene circuit that has 6 transcription units. This is just showing you that each of the genes, and these are core CAM genes, each of these genes is present just using colony PCR. And then these are the number of colonies. So as you can see that as you increase the number of transcription units, the number of colonies goes down, but it's still amazingly an efficient process, Gibson assembly is. So with that, let's get into what these genes do. So these are the subcellular localizations of the carboxylation module genes that I showed you. So this is PEP carboxylase, for example. And the reason that we wanted to confirm their subcellular localization is that you can predict them, but in several instances, we found that the subcellular localization of these enzymes was actually incorrect. And so this was true for PBDK, the PBDK up here, PPCK. This was actually cytosolic, and it was shown to go into, it was predicted to go to the chloroplast. So this is just to show you that it's important to validate the subcellular localization of these proteins with, and these are C-terminal GFP fusions, just to orient the subcellular localization. So now that we knew where these were, we could then combine them into gene circuits. And so this is just to show you, these are expressed under the 35S promoter, and each of the individual genes here is expressed under the 35S promoter. And then we combine them into a gene circuit. So these 4 genes for the carboxylation gene circuit were combined. And interestingly, if you express them together under a circadian, the appropriate circadian promoter, you get a synergistic effect. So this is just showing you that the individual genes don't have much effect in terms of rosette diameter, leaf fresh weight or dry weight, or leaf number. But when you combine them, we see this synergistic effect of a much larger plant, which is shown here by leaf number. And we think that this is because this plant is performing both nocturnal CO2 fixation and daytime CO2 fixation, and it's producing PEP carboxylase, and it's fixing CO2 at night with PEP carboxylase, and it's doing so with all of these 4 genes, and it's able to capture that CO2 into malic acid. And so it increases the carbon gain over any 24-hour dial time period compared to our empty vector control plant over here. So it's basically working the night shift and the day shift, and we expect greater carbon gain from these plants. And this is just showing you that indeed these plants are much larger, a lot more seed set. The seeds are a little bit bigger, significantly. Not a big difference, but it's significant. And so we were very encouraged by this. And so we went on to make the other modules. So this is the carboxylation module that I just showed you. The decarboxylation module, which would take malate that normally the plant makes a little bit of malate, and it would decarboxylate that during the day. And then recapitulate the entire CAM gene circuit. So the carboxylation gene circuit showed nocturnal CO2 accumulation into malate, which is shown here. The decarboxylation circuit showed decreased malate during the day, which is shown here, which we would expect. And the complete gene circuit for CAM shows this decrease and then this increase. So it's basically an additive function of both of these modules into the completed gene circuit. And you can see that normally wild type Arabidopsis, which is the C3 plant, doesn't accumulate malate, but these plants do in a significant way. So how do these perform in terms of drought stress tolerance? So we performed an acute water deficit stress tolerance in Arabidopsis, and we would predict that the decarboxylation module shown here, this would drive down CO2 within the leaf and cause the stomatal to close. And this would cause this plant to be more water use efficient. And so this is indeed what we are seeing with these plants. So this is plants being drought stressed for 18 days and then rewatered. And you can see that the carboxylation module and the wild type don't have much drought tolerance. But as soon as you add that decarboxylation module and the complete CAM circuit, these indeed are more drought tolerant, and the survival rate numbers are shown over here. So what we're working on is to optimize this pathway further, and I'll say a few words about that in terms of what we're doing towards the end. So the last part of my talk, I'd like to just show you what we're doing with tissue succulence engineering. And all of this work I'm going to show you has been published. There are many, these are many CAM-associated traits. CAM plants have many different ones, and these are summarized in a nice current opinions article that we published in 2019. So you can look at that. And these are listed here. But probably the most interesting one for us was tissue succulence. I won't go through all of these others, but you're probably familiar with, tank bromeliads, for example, and in orchids, we have pseudobulbs. There are water storage strategies that these remarkable plants have that we can, make them more water use efficient and allow them to grow in arid environments. The other thing that we're very interested in is epicuticular wax formation in Opuntia, and we have a small project characterizing those as well, but I won't talk about that today. So getting back to succulents. So the reason that we were interested in succulents is that it is a functional trait for CAM. So if you compare the leaf cross-section of a C3 and a CAM plant, they look very different. Many CAM, not all, but many CAM plants have very thick succulent leaves. And the functional importance of this, we think, for CAM anyway, is that this reduced intracellular airspace, which is kind of shown here in these gray parts, is that this is associated with high CAM function. And the reason for this is if you limit the mesophyll surface that's exposed to this intracellular airspace, this will limit CO2 efflux from the site of carboxylation, and it will increase the ability of CO2 recapture by Rubisco during those daytime phases of CAM, phase 1 and 2, particularly phase 2. And this would allow the plant to become much more efficient in terms of recapturing that CO2. So it turns out that CAM, if you look at leaf thickness as a function of CAM, very strong CAM plants have thicker leaves. And this is just data from our orchid project that we published several years ago, just showing you this increase. And it's interesting because weak CAM species have not made this jump to thicker leaves, but they are thought to be evolutionary transition points, or they're on their way to becoming CAM plants, or they are happy being weak CAM plants. It's a matter of debate, obviously, but nonetheless, it's clear that tissue succulence really helps the performance of CAM through this recapturing of CO2. So we were very interested for our CAM project to make succulent plants. And so we were very fortunate to be able to do this by overexpressing a beta helix loop helix transcription factor from wine grape called CEB1. And this turns out to increase, if you overexpress this under the control of a 35S promoter, this will increase leaf thickness by about 40%, increase tissue succulence up to 3-fold, and leaf water content up to about 1.8-fold. So, and you'll also notice that these plants are bigger, and it turns out that this transcription factor will increase the size of all of the plant's cells throughout the plants. So you can see that the plants are bigger. It also produces more leaves, and larger rosette diameters. The seeds are bigger, not a lot, but it is significant, just about 20% bigger, and they produce more seeds. And the reason for this will be apparent in a minute. This also works in Arabidopsis, in tobacco. So you can see that these tobacco plants are a bit larger here. And this is just showing you different parameters. And this is all published. The cell sizes are larger indeed. And the tobacco plants, this is just an example showing that tobacco plants are larger. The roots and leaves are larger. And the reason that this is occurring is that this transcription factor causes more auxin to be made throughout the plant. And this is just shown here with 35S. This is a combination of our succulence engineered plants with an auxin reporter. This is a DR5 or auxin responsive element driving GUS. And you can see that without the succulence gene, the CEB gene, we don't get much auxin reporting, but in this succulent background, we see a lot of auxin. And so this tells us that auxin is being elevated in these plants. And auxin drives a lot of different functionalities, but we investigated this by doing RNA-seq analysis, and we found that over 377 genes were differentially expressed. Their functions are involved in many, many different things. It's quite a complex transcriptome readjustment. And I won't go through all of this, but you can see that here's the plants growing on agar. And so this is our control. You can see that the leaves are much, much bigger. There's a delay in flowering. And again, the leaves are about 40% thicker, so they look like little rosette cabbages. And basically, we saw that many, many genes with auxin-related functions are overexpressed. And these include the biosynthetic genes, as we saw. These plants indeed have increased auxin accumulation, response factors, effectors, conjugators, and transporters. And the net effect of this is very interesting in that we see a differential effect on stomata. And this is very important for the drought tolerance phenotype that we observe. So this is a stomatal closure assay. And basically what we see is a decrease in stomatal density in 4 different lines of our overexpression. The aperture is reduced, and that's shown here in this stomatal closure assay. Normally the stomatal would open wide, but in these plants, the stomatal opening is sluggish. The density, the size is not affected, and the density per plant is not affected. The number per plant is not affected. It's just the density. And why is that? Well, it turns out that we think that all of these cells are larger, and you can kind of tell except the stomata. And if you increase the size of all of the cells within a leaf, except the stomata, you're going to decrease the relative density of stomata within that leaf. So if you combine reduced stomatal density and stomatal aperture, you would expect to improve the water use efficiency of that plant. We also showed that we saw a decrease in intracellular airspace, as I was showing you, and that's going to be important for optimizing the function of CAM. So the integrative water use efficiency of these plants is significantly greater, and the stomatal conductance of these plants is the instantaneous water use efficiency is greater, stomatal conductance is lower, transpiration is lower. Sorry, my mouse is acting up. And interestingly, the CO2 assimilation rate is reduced. This is on an individual plant basis, but because the plants are larger, there's no downside to this at the whole plant level. In fact, the plants are larger. So are these plants indeed more drought tolerant? Well, we did, again, an acute drought tolerance assay, and looking at these 4 different lines, before, during, and after water deficit stress for 14 days, we can see that these plants do a much better job of surviving this drought stress. And this is just 14 days. You can see we kill our control plants for the most part. But we have lines, some of these lines will survive for 30 to 60 days, which is really amazing. And it's all attributable to that just little increase, that doubling of the onboard water that these plants have. So their survival rate in instantaneous water use efficiency. So the other thing we looked at is salinity tolerance. And we thought, well, we should look at this because what if this succulence is diluting the ion content of these plants? Well, it turns out that that hypothesis is actually true, that these plants are more salt tolerant. And this is just shown here, watering these plants for a month at 250 and 300 millimolar. And you can see that these plants are quite nicely surviving. The survival rate is shown here. And it could be a combination of dilution, but also reduced sodium uptake. And this is just showing you that it's probably dilution. So this is the sodium and the chloride content of the leaves. We did a large ionomic profile. And if you're interested in other ions, these are all reported in the paper from 2020. I won't go into all of those data, but this is the important sodium and chloride. But we saw many other changes as well. And so this is quite interesting that this transcription factor is impacting the ion accumulation of so many different ions. So it turns out that not only do we think there's a dilution effect going on, but these plants also appear to take up less sodium through changes in their sodium channels in the roots. And so this is just using a sodium detection fluorescent probe called Corona Green. And you can see that at 50 and 100 millimolar salt, these plants have much lower sodium uptake compared to our empty vector control. And again, if you'd like to know more details, this is all in our paper. So with that, I'll switch to our future directions and just tell you where we're headed with this research. So we're continuing our work on Opuntia, and we're continuing to look at productivity of different accessions. We're looking at other production barriers, like there's a stunting disease, there's cochineal insect pests and other issues associated with Opuntia production. And then we're continuing with our CAM engineering work, and we're working on optimizing different aspects of CAM by looking at malate transport, carboxylation pathways, and starch degradation pathways. And we're also determining whether, if we combine CAM with tissue succulence, does this improve the efficiency of CAM? That's really the big question. So here's this slide again, just to show you that ultimately we are targeting soybean, but we're also looking at optimizing the decarboxylation module and the starch pathway. And turns out that CAM plants degrade starch using the phosphorylytic pathway compared to a C3 plant like Arabidopsis, which uses the glycolytic pathway. So with that, I'd like to acknowledge most of the work that I've shown you was conducted by all of these people, but just point out Durba Nupani, who did the Opuntia work, Dr. Yim, who's our bioinformatic collaborator and former postdoc in my lab, and Dr. Lim, who also was a former postdoc in my lab, and he's now a professor in South Korea. And Dr. Yim is a professor in our department. And with that, I'd like to acknowledge the work. Our early CAM work was funded by the Department of Energy. Our current project is funded by the NSF, and our Opuntia work is funded by the USDA NIFA. And of course, we get a lot of support from our own agricultural experiment station and the Joint Genome Institute, who've sequenced many of our genome and transcriptome projects. And with that, I will go ahead and stop sharing and take any questions.
Thank you very much, John. So if you can make the questions, Joseph?
Yes. Yes, hello. That's John. Thank you very much. Hello, and that's really amazing work you do and very nice talk. Thank you very much. My question is related to this analysis of the accessions of the Opuntia that you do. Are you looking to the roots?
We're not looking at the roots per se. We're just focused right now on the chloroplast genome, but the way that we are sequencing those chloroplast genomes, we're actually sequencing a slice of the chlorancoma from the cladode, and we're using Illumina. So we actually have about 25 million reads for every one of those accessions. Those data are used to assemble the chloroplast genomes, but we have a lot of nuclear DNA reads as well that could be used for other studies like GWAS.
Okay, okay. And another question is related to auxin. So there are some cases when you have very high DR5 activity, but zero auxin. So when you see this high auxin response, did you check auxin concentration?
Yes, we did. And that's in a paper. We measured auxin directly to be sure. Yeah.
Okay, okay. So there is a correspondence, I guess, right?
Yeah,
and in general,
agreed nicely.
Okay. And in general, what is the mechanistic link between CAM and auxin metabolism?
Yeah, none that I know of. So auxin is simply creating this, producing increases in cell size, and it also increases the apical, the number of shoot apical merostems and also root merostems. It proliferates those apical merostems, and it increases cell expansion. So that has nothing to do with CAM per se. So we don't know if there's what the interaction of this will be with CAM, although that's something that we're going to be looking at, because we're going to combine the two. We're going to combine the engineered CAM with the tissue succulence and see what happens. Yeah,
so it's simply my thoughts are that it's kind of an internal contradiction, because you have high auxin, high root biomass, and low and high water use efficiency, so you don't need too much roots, apparently. So I just want to understand this relation. Do you have some thoughts about that? Because you you mentioned there is a root biomass is also increasing.
Yes, it is. Yeah, everything is larger. So all tissues, whether leaf or root, are similarly enlarged in these plants.
Okay.
And but because of that closure of those stomatal apertures and the reduction in stomatal density, we see a higher water use efficiency that I think is disconnected from that root phenotype.
Okay, okay. Very interesting work. Thank you very much.
Thank you. Other questions?
Any other question? Miguel?
Thank you very much for your seminars. Very interesting. When you say that the seeds are bigger, this is because they contain more starch or more stored proteins, which is the that makes the seeds bigger?
Yeah, we didn't look at that. We looked at protein content within the leaves, and that is higher. But because the plants, they make more chloroplasts per cell. That's in the 2018 paper. But we did not look at the seed composition per se, and that would be something nice to do in the future.
And the other question is, these bigger seeds germinate in a high percent, or how is the percent of germination of these bigger seeds?
It's the same. We didn't see any difference. We did do germination assays with these seeds under various osmotic conditions and salt conditions, and that's all published in our 2020 Plant Journal paper. But they didn't show, for example, a germination, defect of any kind, or an advantage. They were the same as our wild type seeds.
My last question is, you are increasing the amount of carbon into the plant, and it is clear that there is a balance between carbon and nitrogen. These plants could be better nitrogen assimilators.
Yeah.
What is your opinion about this?
Yeah, perhaps. They're obviously being grown under nitrogen replete conditions. We're not limiting nitrogen, so you would have to do a nitrogen limitation experiment to sort of try to get to that answer. Again, something to do for the future. But yeah, nitrogen use efficiency and CAM is a really interesting question. Some have suggested in the literature that CAM plants are more nitrogen use efficient because they have to make a lower investment in rubisco, but because the PEP carboxylase is also made. I don't know. It's an open question. I haven't seen anything that really is definitive in that regard.
Thank you very much.
Thank you.
Thank you. Claudia?
Thank you. Yes, fantastic work. Thank you very much. My question is about these bacteria that digest Opuntia, this Pectobacterium cacticida.
Yeah.
Pectobacterium is a pathogen, yeah? And so how do you expect to use it? I guess just in a closed system, not or anything like that?
Yes, we would not want to spray the field with this bacteria because it is, as you rightly point out, it is a pathogen. It's a recognized pathogen for Opuntia, actually. It's been studied in that regard, and that's all summarized in our paper. Yeah, so it would only be in a bioreactor setting.
But you isolated it from the field, yeah? So it
yes, we isolated it from the field, but it was in the soil and it was in rotting pads in the soil. So what they do, this is from a commercial plantation in Salinas, California. What they did was they took the pad trimmings, the cladoed trimmings, and they would just put them on the ground, and then they would rototill them in, and sort of as a disposal of that biomass. So we scooped up those degrading pads, and that's the material with some that were in the soil, and they were nicely decaying, and we grabbed that, and that's what we characterized.
Yeah, okay. Thank you very much. Yeah, I was just wondering if it is more common to find it in other places where it could be dangerous to find it.
Yeah, it's not a big problem. The fungal diseases, are also a problem, but we also study Opuntia stunting disease, which is a bigger problem, and of course cochineal, which is probably the largest problem for Opuntia production. Yeah.
Okay, thank you very much.
Okay.
Yeah.
Okay. Who else? Alejandra?
Yeah. Hi, John.
Hello.
Very nice talk. Interesting. How are you?
I'm good.
Yeah, I have this question first about the genome size of this cacti of Opuntia. You said that it's a very large genome, and I wonder whether which are the genes that are more reiterated in this kind of in this species, if they are related with this CAM metabolism or any other of processes that seem to be that have helped to adapt them to these harsh conditions.
Yeah, don't know. We're digging into those data. I can't give you an answer at this time, but we did do a metabolomic study this year that we published earlier, and I can send you that. It doesn't. We don't see any from the metabolomic profiling. We don't see anything that's too unusual. That's sort of unique, something that stands out. So in terms of stress adaptation, Opuntia seems pretty normal. So I think it's relying on CAM heavily for its adaptation, rather than other, either osmolytes or other Lea proteins, for example. But we'll certainly be looking at that very closely as we go through the genome annotations.
Okay, thank you. And another question is regarding this transcription factor that you overexpressed in Arabidopsis to get these succulent characteristics. And so do you know which are, in Arabidopsis, which are the targets of this transcription factor to have such a pleiotropic and very well adjusted characteristics in a plant that is far from being a succulent? You get it?
We have RNA-seq readouts. We haven't done ChIP-seq on these plants to look at binding sites, but it's a really diverse collection of genes, and we studied them quite closely, and those are reported in both of our 2018 and 2020 papers, and it's very complicated. many genes, many hundreds of genes are being affected by this transcription factor, and but auxin seems to be the major driver of the succulent phenotype. That's very clear.
But to get this expansion in the cells, you mean of course auxin is involved, but also to get water inside the cell, you would need something else, right? So like a very good osmotic adjustment and things like that. So I suppose you have seen that also in Arabidopsis.
We have. So that's in our 2020 paper. We looked at all the major transporter families, and we saw number of potassium and sodium transporters being affected. And those are probably contributing to particularly potassium is going to drive cell expansion. So we do see evidence for that. Yeah. And we discussed that in our 2020 paper.
And the last question. So do you think, well, I suppose also that the hydraulic driving force is changing these plants, right? So once can be the osmotic adjustment that must cells having these conditions, but also what about the stomata? I mean, it should be some driving force produced from the stomata to take water from the roots and
yeah, well, perhaps, but that doesn't seem to be so important. But obviously there's enough transpiration, although it's very reduced in these plants to drive water uptake. So they don't have an impairment in water uptake. And so it's having these sluggish stomata doesn't seem to impair that. And this has been demonstrated in wheat as well, where if you reduce the stomatal density in using another way, what happens is that the plants have greater water use efficiency, but phenotypically they look normal. They don't seem to be impaired in dry, that water uptake. So there doesn't seem to be a negative trade-off,
so maybe
at least at the levels that we've changed stomatal density in these plants.
Right. So maybe the driving force is more the pulling up water.
Yeah. Could be.
Okay, so thank you. Nice talk.
Thank you.
Thank you.
Good to see you.
Any other question?
Helena.
Elena. Elena, tu micrófono.
Very nice work. I enjoy it very much. But I would like to ask you about if you can compare the obtention of biofuels through Opuntia with algae cultures, for example, that doesn't use land and is supposed to be interested to get biofuels through algae cultures.
Algae.
Yes.
Yes. Okay. So we used to work on algae, and I don't work on algae anymore because I don't think that economically algae is a viable option. The major reason for that is that the capital costs of setting up algae ponds and harvesting algae are just too, it's just too expensive for the return that one would expect to get from algae. And so we can plant Opuntia at very low cost, almost nothing.
Yes, a lot of land for that.
And then you have a crop, and all you have to worry about is harvesting it, which is pretty straightforward. It's much more difficult to do so with algae.
And you are not going to use very much water for that. So
that's right. Whereas with algae, you will be replacing the water in your ponds like every month because of evaporative losses.
Yes.
So I studied halophytic algae for a number of years. And once I realized that it's economically not going to work out, I dropped that and moved on. So I think Opuntia is a much more appealing option as a feedstock.
Very interesting. Thank you.
Yeah.
Mm-hmm. Any other question? I have one, John. Do you see a relation between the plant size and the ploidy numbers that you show?
Yes.
So that plant that's 20 times ploidy, that has to be huge.
No. that relation. I thought you were asking about Arabidopsis. No, we do not. We do not see we do not see a relationship with those really large ploidy levels. Of course, if you compare a diploid coccinifera with an octoploid coccinifera, there's a big difference. I would say there's like a 4-fold increase in cloud size. So there's definitely a difference there. Yeah. But
Okay.
when you go to 20x chromosome number, they look, anything above octoploid, they look like an octoploid. There's no difference.
Very different. Okay. Well, you have answered several of the questions I wanted to ask about the transporters, and I guess I have to look at that 2020 paper to get more information. Yeah. Well, if there is no any other question, I would like to thank John for the very exciting, very interesting talk, very stimulating research, and I hope you have enjoyed. John, thank you very much for your visit, virtual, and hope to see you sometime.
Yeah, thank you so much for the invitation, and I'm just sorry I couldn't make it in person, but hopefully in the future.
Yeah, we will try to bring you down.
That'd be great.
Thank
All
you
right.
very much.
Well, thank you so much. All right, take care, everybody.
Yeah, thank you.
Bye.
Okay, John. Well, thank you very much. Thanks, John. And now, just last thing, if you could send me some of those seeds you mentioned, the mother cell mutant.
Yeah.
Okay, you send them by normal mail?
Yeah.
You mind?
can do that.
Yeah.
Send me your address, and I'll.
I send the address, and
I'll get them to you.
Okay. Yeah. Okay. Well.
Okay.
Thank you, Ryan. John, thank you very much. Very nice work you are doing.
Okay.
I hope to see you sometime